Hello, my name is Nino Chiocca. I'm the professor and chair of the Department of Neurosurgery at the Brigham and Women's Hospital and Harvard Medical School. Thank you for allowing me to give you this first look. Our first look today is on oncolytic viruses, how we turn pathogens into anti-cancer agents. These are the problems we are trying to solve. For several highly lethal cancers, new therapies, such as targeted therapies, have not been successful. Two of the most lethal cancers are shown on the scans on the right. One is glioblastoma, the other one is pancreatic cancer. The question is, why have targeted therapies and immunotherapies not been successful? And there are two problems that underscore this lack of success. One is intratumoral heterogeneity. What does that mean? That means if one takes biopsies or samples of different sectors of each of these tumors, one will find is that the tumor cells, once you sequence them, will have several mutations. But then you will also find that tumor cells taken from another sector may have completely different set of mutations. Therefore, if you have a targeted therapy against one set of mutations, it may not work against the other set. And if you reiterate this over and over again throughout the tumor, this shows you why single or even combination agents have not worked. Why has immunotherapy not worked? Immunotherapy has not worked because these tumors are also known to be immunologically cold. What that means is that there's very few, if any, inflammatory anti-cancer immune cells within the tumor. Our solution is based on oncolytic viruses. These are viruses that have been genetically engineered to infect and spread in tumoral but sparing normal cells. Why are oncolytic viruses a solution to intratumoral heterogeneity? When one looks at cancer, cancer has to basically perform several phenotypes, or is characterized by several phenotypes, in order to grow and progress in an organ. Each one of these phenotypes is the accumulation of multiple mutations. But there are only very few phenotypes shown here, while lots of mutations. Turns out that viruses and cancer cells both thrive in the same biological phenotypes and environments. Viruses can grow in the same phenotypic areas that tumor cells do. Why are oncolytic viruses a solution for immunological coldness? That is because if one takes a tumor like a glioblastoma, which we said is very immunologically cold, if you add to it an oncolytic virus, the body will react to it by infiltrating it with lots of anti-tumor cells called your CD8 plus T cells and CD4 plus T cells. In our laboratory over the years, we have generated several oncolytic viruses based on herpes simplex virus type 1. This is shown here as OHSV. We've had multiple preclinical experiments with various types of oncolytic viruses. And then I brought one in particular to a phase 1 trial for the highly lethal malignant tumor glioblastoma. We have shown this virus is safe. And in this figure here, you can show that there's oncolytic virus presence within the tumor after injection. And there are also lots of T cells that are infiltrating into the tumor, just as what we hope we will get. Our newest invention now is a next generation oncolytic virus. We've now made two changes to our previous version. One change is to remove a gene from the virus that is highly virulent. In other words, it causes some of the safety and toxicity issues. This gene is called ICP34.5. But this gene is also needed by the virus to really replicate and biodistribute well throughout the tumor. We've substituted this with our homologous gene. It's actually a human gene called GAD34 that exhibits no neurovirulence and actually provides even more viral replication. Who will benefit from this invention? This oncolytic HSV1, which we've called GAD34, is applicable to highly lethal solid tumors via an intratumoral route. For example, there are approximately 20,000 glioblastomas and 40,000 pancreatic cancers, and many more other solid tumors in the USA per year, which this could be applicable to. What is next? We are looking to commercialize this GAD34 oncolytic HSV by licensing it, forming a new company, or by partnering. Ultimately, the road to IND-enabling studies and to start our first in-human phase 1 clinical trial will require 2 to 3 years and likely $2 to $5 million. Thank you for your attention.